Hey everyone, today we're going to start chapter 3, and this is going to be about cells and tissues. We're going to talk about cells today, and most of this should be a review from biology, so we're going to cover quite a bit today, again, because it should be a review. If you're struggling though with the material, please make sure that you're asking for help. We're going to start off by asking what are cells, and cells are defined as the structural units of all living things. There are trillions of cells in the human body, and there are more than 200 different types of cells that vary pretty big based on size, shape, and function. Now, the reason there's so much cell diversity is because the shape of the cells and how many organelles are located in the cells is going to be relative to what is that cell's specialized function. Are they like a nerve cell that need to transmit a response? Are they like our muscles that need more mitochondria in order to produce ATP? So they're going to be different based on what they're trying to accomplish. No matter what, cells are going to have the ability to metabolize. In other words, they're able to create chemical reactions, digest food, dispose of any waste that they create, reproduce, grow, move, and also respond to a stimulus because cells are considered to be alive, and these are the characteristics of being able to live. From here, we're going to start talking about the anatomy, or what the structures are, of a generalized cell. While no one cell type looks exactly like the others, there are some basic parts that all cells have in common and that serve certain functions. So in general, the cell is going to have three main regions that we're going to talk about today. The first is nucleus, then we have the plasma membrane, followed by the cytoplasm. We're going to begin by talking about the nucleus, which right here in this image is going to be purple. The nucleus, the whole function is to be the control center of the cell. It is going to contain our genetic material that we call deoxyribose nucleic acid, or otherwise known as DNA. And DNA is very important for building proteins, but also this DNA is passed down whenever our cells reproduce or our cells are going to create offspring. The nucleus has three regions. It has a nuclear envelope. There is an area called the nucleolus, and then there's chromatin within the nucleus. So let's talk about each of these three regions. But first, here's an image of your nucleus. Here's our nuclear envelope. Here is our chromatin labeled, and then our nucleolus is this dark center. So first, talking about the nuclear envelope, this is actually a membrane that goes around our nucleus. It has pores in it. So here are an example of our pores that are in our nucleus. And these pores actually allow for materials to be exchanged between the nucleus and the rest of the cell. Within the nucleus, there is a fluid called nucleoplasm as well. Section 2 is the nucleolus. This is more of a darker stained part of the nucleus when we're looking at a slide. And the important part about the nucleolus is this is where ribosomes are assembled. Now, ribosomes are an organelle that we're going to cover later in this lesson, but the whole point of a ribosome is eventually to go make a protein. What happens is once ribosomes are made, they actually go out through the nuclear pores into the regular cytoplasm, again, where proteins are going to start to be made. The last thing is chromatin, and chromatin is actually made up of DNA, but it's just DNA wrapped around proteins called histones. This is all throughout the nucleus, and it's really hard to see when the cell is not dividing. Whenever the cell is dividing, though, they tend to condense to form something called chromosomes whenever the cell divides, which should sound like a familiar word when you guys talked about mitosis in biology. So if we're looking back at this image, here you have the nuclear envelope, chromatin spread all throughout it, and then again this dark middle region is your nucleolus. From there, the second thing most cells have in common is your plasma membrane. This is a transparent barrier that contains cell contents. It separates cell contents from the surrounding environment. So again, it maintains boundaries. It is made up of a biolipid, and it's a biolipid of something called phospholipid. phospholipids. It's also made up of cholesterol, proteins, and sugar groups. And one thing to know about the plasma membrane is that it is considered semi-permeable. What that means is it allows some things in and some things cannot enter into the cell. So here's an image of the cell membrane. We have a double layer phospholipid membrane. So here is one phospholipid, here's the other. So it's two layers. It has proteins in it, such as right here. We also have lipids in the structure. We have cholesterol. So multiple things make up your cell membrane. From there, we're going to talk about three main types of cell junctions that we're going to see within the cell membrane. And when I say cell junction, this is just how cells are bound together. The first one is tight junctions. These are called tight junctions because they are impermeable junctions. What that means is that they are leak-proof. They are bound so tightly together that they do not let anything in or out. Plasma membranes usually are going to fuse like a zipper, again, to prevent substances from passing through them into the cell. The next type of cell junction is called a desmosome. This is going to be a cell junction that anchors things. So this helps whenever the cell is being pulled by mechanical stress, anchor it to where it's attached to. Often you're going to see these in tissues. They like to be anchored in place within your body. These are created by button-like thickenings of adjacent plasma membranes. And the last one is a gap junction, which is kind of the opposite of a tight junction. These are sometimes called communicating junctions because they allow communication in substances to move from one cell to another. And typically, they are connected through proteins called connexons. So again, molecules can travel directly through this gap junction to another cell. So here is an example of these different types. Here is a tight junction. It's impermeable. It's kind of like a sheet. It doesn't let anything in or out. Our desmosomes, you can see these little wires off of it that help anchor it down. And the last one we have over here is this one, which is a gap junction. Notice it's going to allow you to go from one cell to the other. So here we have substances going from this cell over to this one. And last but not least, we're going to talk about the cytoplasm, which is the biggest section. This is going to be all the material outside of the nucleus, but inside the plasma membrane. It is where almost all cellular activities are going to take place, and it's made up of the cytosol, inclusions, and the main part, organelles. So the three components, cytosol is just fluid that contains a bunch of nutrients and electrolytes. Inclusions are any chemical substances that are stored within the cytosol. So this includes things like nutrients and other cell products that kind of float around. And the main steal of the show is organelles. This is the machinery of the cell that performs its function. A lot of these organelles are considered to be membrane bound and they compartmentalize and have specific functions. So here's an overall image of the cell. And again, so far we've also talked about cytosol, which is the fluid. We have talked about the cell membrane or the plasma membrane over here, which is going to be the outermost part of it. But now we're going to talk about these things kind of in the middle. So things like mitochondria and centrioles. So let's get started. Mitochondria is first. Mitochondria is made up of a double membrane with cristae on the inner membrane. But what you guys probably have heard, it is the powerhouse of the cell. And what that means is that it is going to be the part of the cell that carries out reactions where it takes oxygen and it uses it to help break down food to make ATP molecules. And ATP, if we remember from biology, is energy. Ribosomes are another type of organelle. Ribosomes is where you're going to see protein be made. This is the site of protein synthesis in the cell, and you can find ribosomes in two locations. You can find it free-floating in the cytoplasm, or sometimes you can find them attached to something called rough endoplasmic reticulum, or sometimes we abbreviated this as ER, so our rough ER. So what is endoplasmic reticulum, or ER? These are fluid-filled tunnels or canals that carry substances within the cell. They are typically continuous with the nuclear membrane, and there are two types, rough ER versus smooth ER. The main difference from them is, do they have ribosomes attached to them or do they not? So our rough ER is considered rough because it's studded with ribosomes. Again, this is where protein synthesis takes place. And what happens is once they synthesize these proteins, transport vesicles, which are just kind of like the mini cars of a cell, move these proteins within the cell. And these are going to be found very abundant in cells that have to make and export a lot of proteins. Your smooth ER, this is smooth because it lacks ribosomes. And the whole function of the smooth ER is to have lipid and steroid synthesis. Whenever I say lipid, typically we're going to be talking about fats. So I know that's probably not a word you guys are familiar with, so lipid just means fats. The smooth ER also is very important for detoxifying drugs and any kind of pesticides that could potentially get into your body. Next, we have the Golgi apparatus. This is a stack of flattened membranes. It's usually associated with tiny vesicles. So again, remember, vesicles are like the cars to transport things. What happens is it takes these packages that it gets from the ER and it modifies these packages and actually sends them out to where they need to go. So again, modifies and packages any kind of proteins that are coming from our rough ER and sending them to where they need to go. Do they need to go to another part of the cell? Do they need to be exported and sent out of the cell? That's what the Golgi is going to be in charge of. Then there are some things the Golgi decides we don't need anymore, and she might send them to something called lysosomes. Lysosomes are going to be membranous bags that have digestive enzymes. So whenever a cell has parts that wear out or things that they don't need, it's going to be engulfed by a lysosome, and the enzymes in it are going to break down that structure into things that we're able to use. This also includes any bacteria or debris that get in the cell. They let out some phagocytes, which is a type is going to dispose of things. Next, we have peroxisomes. I don't think this one is covered in biology, so this is one that might be new, so I'm just going to go ahead and start it. Peroxisomes are membranous sacs that contain something called oxidase enzymes. And their whole function is to help detoxify any harmful substances like alcohol or formaldehyde. And it does this through breaking down them, any free radicals they might have. So free radicals are typically highly active chemicals. And what they do is they take these free radicals and they convert them to hydrogen peroxide, which then breaks down into water. Hence, hydrogen peroxide peroxisomes, where they get their name. These typically replicate by pinching in half or budding from the ER to create more. Then we have the cytoskeleton, exactly what it sounds like. The skeleton is going to be the framework or the network of structures that extend all throughout the cytoplasm that gives the cell some type of internal framework. It's going to determine the cell's shape, support any organelles that's in the cytoplasm, and then also allow basically the roads that things get transported along. There are three types of elements. There are the microfilaments, which are I have this backwards. The microfilaments are the smallest. You have intermediate filaments, and then your microtubules, please change to be your largest. So again, you have microfilaments, which are pretty small, and you have microtubules, which are generally pretty big. And then we have our centrioles. Centrioles should be a term that sounds familiar because we talk about them when you guys go through cellular reproduction or mitosis. And these are rod-shaped bodies made of about 9 microtubules. And these actually make the spindles or the mitotic spindles that our chromosomes separate on during metaphase and anaphase. that occur during cell division. So typically in cell division, the centrioles go to opposite ends of the cell, and they release their little spindles, again, for those chromosomes to travel along. Last but not least, we're going to actually end this section talking about cell extensions. There are some cells that have surface extensions found on them, and the three that are most familiar are cilia, flagella, and microvilli. Cilia tend to be located on the surface, and these are basically small little extensions that help move things through. So, like, for example, you're going to find them a lot in your respiratory system to help move mucus. So, again, if you have the cell, they have little projections off of it, and they kind of swim back and forth. Then you have your flagella. This is typically going to be, if you had a cell, it's going to be one long projection, and this is usually found in sperm. And then you have microvilli. These are even tinier than cilia, and these extend off of cilia typically. And they also, the whole point of them is to increase surface area for absorption. So you're going to see a lot of microvilli within your digestive tract. And that is the end of today's lesson. I know it was long, but most of it should have been review. If you have any questions, please just make sure that you ask. And I hope you guys have a great day.